Well, hi, my name is Juan Miguel Machare and my topic is about exome sequencing and the management of neurometabolic disorders. The definition of this topic we divided into parts: exome sequencing and neurometabolic disorders. Exome sequencing is a technique for sequencing of the expressed genes in a genome known as exome. And neurometabolic disorders is various disorders that result from genetically determined abnormalities of enzymes, the metabolic sequences of which affect the development or functioning of the nervous system. Although most occur in children, some can present in adult life, and increasing number of affected children survive into adult life.
The human body is made up of 46 chromosomes that come in 23 pairs, one set from our mother and one set from our father. Chromosomes are like different books in an encyclopedia set. Each book carries different information, yet altogether they make up a very important reference set. Our chromosomes are like these books in the encyclopedia set. If one is added, deleted, or out of place, it may cause disruptions in the reference information. The severity of this disruption may depend on how much information is changed as a whole. If we take a closer look at each volume of the encyclopedia set, we will notice that there are pages upon pages of information in each one of these books. These pages are analogs to the genes that code for everything in our body, from tissues to blood to different bodily functions. If these genes are misspelled, misplaced, or moved to a different location within our genome, it can have mild to severe effects on a person's health. But how exactly does this happen? Just like the words on the pages of the encyclopedia, our genes are made up of 4 letters: A, G, C, and T. These 4 letters create the genetic code. In our written language, we expect the letters in words to be in a certain order for our brains to translate the correct meaning. If a letter is added, out of place, or deleted, the word itself may not be understood. Our genetic code works along a very similar translational system. A gene is made up of letters that need to be in a specific order for healthy proteins to be made. If a letter is out of place, added, or deleted, it too may not be understood by the human body. We call this a genetic alteration or a mutation. Some genetic alterations are considered benign or not harmful. These spelling changes in our genes may even be beneficial as they introduce genetic variation in our species. Benign changes may be the cause of humans having different eye color, different hair color, or even different personalities. However, if a genetic alteration is severe or in a part of a gene that has high functional value, it can be the cause of human disease or death. Many times, we call these harmful spelling mistakes in the gene mutations or pathogenic alterations. We are learning more and more about the causes of these spelling mistakes on a daily basis. It is not always clear why some people carry these gene changes while other people do not. In fact, many gene changes happen sporadically in people with no family history of the same change. This can often be difficult to diagnose and can sometimes be confusing for the patient, as they often wonder why this has happened to them. In the past, clinicians have been able to detect these spelling mistakes by sequencing suspect genes one at a time. This often resulted in a costly and time-consuming process. With the advent of clinical exome sequencing, clinicians are now able to simultaneously survey all 20,000 genes. Clinical exome sequencing allows clinicians to better treat and manage their patient's disease by helping them understand the underlying genetic cause. The human body is a complex system, and there is still so much more to be understood. As researchers and doctors continue to learn more about the human body, we will also learn more about how to create better treatments and cures for genetic conditions.
On application in medicine, we have that the next generation sequencing have revolutionized the discovery of genes in which variants cause rare Mendelian diseases. A diagnostic yield of 60% has been documented for whole exome sequencing among patients with unexplained intellectual development disorder, a condition that affects an estimated 3% of the population worldwide. along with coexisting illness that include epilepsy, psychiatry or behavioral disturbance, movement disorders, sensory deficits, and other organs dysfunction. Intellectual development disorder poses a substantial emotional, functional, and economic burden. Copy number variance, methylation abnormality, and single gene defects are known to cause intellectual development disorders. On pros and cons, we have that on pros, we have two aspects. That first is the high coverage in target regions, and second, to reduce the cost of large genomes in people. And in cons, we have two aspects too. The mutation in regulatory regions will be missed, and for second, the genetic markers can only be genotyped if they are in the target regions. And for another point, we have the issue that we apply in Ecuador. In this video, we have an explain of that.
Metabolic disorders are caused by an abnormal metabolic process. It can be congenital due to inherited enzyme abnormality, or acquired due to disease of an endocrine organ, or failure of a metabolically important organ, such as the liver. Inherited metabolic disorders are caused by a defect in a single gene. There are many inborn errors of metabolism. Some produce relatively unimportant physical features or skeletal abnormalities. Others produce serious disease and even death. Some of the more familiar inborn errors of metabolism are cystic fibrosis, hypothyroidism, sickle cell anemia, phenylketonuria, PKU, and Tay-Sachs disease. Symptoms of metabolic disorders: body fluids that have a maple smell, bone abnormalities such as osteoporosis, difficulty with memory, thinking, talking, comprehension, writing or reading, enlarged liver, heart, kidney or spleen, failure to thrive in infants and children, frequent infections, hypoglycemia, low blood sugar, loss of vision or changes in vision, muscle twitching, spasms or seizures, muscle weakness, paralysis. Treatment for metabolic disorders: bone marrow transplantation, medications to reduce symptoms, such as pain or low blood sugar, mineral supplementation, nutritional counseling, physical therapy, surgery to relieve pain or symptoms, vitamin supplementation. Metabolic disorders is a common disease, yet every individual needs unique treatment and care. We encourage people with metabolic disorders and their families to learn as much as possible about the latest medical treatment and approaches, as well as healthy lifestyle choices. For more information about metabolic disorders and how to treat metabolic disorders, just click on the link in the description or visit www.mainmd.com. Thank you.
And in a conclusion, I say that whole exome sequencing has transformed gene diagnostic in rare diseases. Translation into disease-modifying treatment is challenging, particularly for intellectual developmental disorders. However, the exception is inborn errors of metabolism. Since many of these disorders are responsive to therapy that target modification pathology features at the molecular or cellular level. And I finally point, this last video gonna tell you as a summary of what my presentation was about.
MDA just announced a new $400,000 infrastructure grant to Brian Traynor at the NIH to sequence the exomes of 1,000 ALS patients. So the exome exome is a small part of the genome. You've probably heard of the Human Genome Project, where they're sequencing the whole of the human genome. The exome is the small part of that genome that codes for proteins. So it's the parts of the DNA that does something that we know about. That's not to say the other parts of the genome don't do anything, but most disease mutations are part of the exome, which is a much smaller part of the genome than the entire thing. So we're going to sequence just those parts because that's where we're most likely to find new disease mutations. So Trainer's project is particularly interesting in the sense that they're not only sequencing 1,000 ALS patients, the majority of those patients already have donated tissues that are in a data bank. So not only will all of the information from this project be put up on the internet and available to researchers around the world, if they come up with a new hypothesis, they're going to be able to get hold of the tissues and follow up their hypotheses by actually testing those hypotheses, really seeing what's going on at a tissue level. So it's going to be really exciting because this isn't just going to help the Trainer lab, it's going to help researchers all around the world to look at the data from this project, get the tissues associated with those samples and follow up on those hypotheses. And we're really hoping that that data, once it's available at the end of the year, will really help us move ALS research forwards. So right now we're in a really interesting place with ALS research in that we still don't really understand what causes the disease. About 10 to 15% of cases are familial, they're inherited through the genes, and the rest seem to be sporadic. We're beginning to find some new mutations which cause the disease. And a project like this is going to be really exciting because we're going to be able to identify new gene mutations that cause the disease and possibly modifiers of that disease, which means you may have a patient that should progress very quickly and that patient doesn't. We may be able to find out what's different about that patient and therefore get some really new and exciting ways of treating the disease. So this is the main goal of this, is to identify new genes and new mutations that can cause ALS, but it's going to tell us a lot about the fundamental causes of the disease.